Welcome to Cold Spring Harbor Laboratory Live at the Lab. I'm Aline Augenbraun, and it's a great pleasure to introduce Partha Mitra, who is the Crick-Clay Professor of Biomathematics at CSHL. He also has a distinguished chair in Computational Brain Research at IIT Madras. So Partha, you and your colleagues just published a paper in Nature Machine Intelligence. You taught computers to understand brain anatomy, cell by cell, better than any previous machine learning tool had ever done before. You were able to trace individual neurons meandering through a field of a bunch of cells, whatever they were. And that's something that humans do well, but machines do poorly. So congratulations for that, but tell us, why should we do this? Why should we teach computers to do things that we do well already?
Well, because the data sets are very large. So we are trying to understand these neuroanatomical connections in brain circuitry, because that's how the brain works. The distributed algorithms that run the brain are instantiated in the circuitry. And to map those circuits, which has previously been done by a neuroanatomist looking carefully under a microscope at stained sections, and that's been going on for over 100 years now. That method isn't really scalable. So as we are gathering a lot of data, and the data sets have gotten really large. So if you think of the human brain at light microscope resolution, that's about a petavoxel worth of data. And just in terms of scale, I understand that, all the photos on Facebook, that's a few hundred petabytes, of data, and maybe 1,000 petabytes. And so a human brain being a petabyte, you start studying 1,000 human brains, you're already going to reach enormous scales. And you can imagine, unless everybody took a degree in neuroanatomy, we won't have enough neuroanatomists and enough microscopes to look at all those images. So if we want to study those in a scalable way, we really need assistance from the computer. So we are trying to automate what the neuroanatomist expert does, so that we can ultimately achieve our goal of studying these circuits, understanding how they work.
So why do we need to understand the circuits? What will we gain by understanding our brain circuits?
Well, I mean, there's the pure curiosity of how it works. There's obviously the technological implications that are now writ large. People are trying to make intelligent machines, and there's at least one line of thought that says that understanding our own brain circuits better will help us automate better. And there's, of course, the medical side of things, where if you have a neurotypical brain versus something has gone wrong in the brain, well, something has materially changed in the circuitry, and understanding what the change is will help us understand what to do about it. So, from medical, technological, and I think purely intellectual reasons, the last one is the one which personally I find the most motivating, the task of understanding ourselves.
It's always important, but so I can't help but think, like children learn this so easily. They learn how to see two things that look disconnected and see a string between them. They know that they're connected. But computers have a hard time with that task. Can you tell us a little bit about how you trained the computers to see these connections?
Well, the specific thing that we were focusing on in this paper were tracing fragments of neurons, axons, dendrites, etc. And one of the difficulties with the machine learning algorithms that are currently in vogue, the deep nets in particular for this kind of image processing task, is they utilize local rather than global information. So connecting two dots over a long distance is kind of a global task. And just empirically, what we found was that the best-in-class algorithms, which we applied to the problem, sometimes would fail at that. And so we introduced some topological priors which captured that kind of global information. How human neuroanatomists do it, or I'm sure I could teach a school child, a kindergartner, to do this very easily. because you see it and you know it. How we do it, that I don't think we still yet quite understand. I mean, there's been a lot of classical work in vision, about completing lines, and I won't go into all of that. But it is certainly true that when we took the state-of-the-art machine learning algorithms, they weren't performing quite at the level we needed them to, so we needed to augment it.
So explain how topology works. Should I put up the slides so you can talk people through how you use this clever math trick to help the computer? So I'm going to share screen. So hold on a second.
The topology often is referred to as rubber sheet geometry. So imagine drawing a figure on a sheet of rubber on a balloon and then blowing up that balloon. The properties of the figure that do not change when you do that are topological properties. The geometrical properties will change. So the lengths of lines will change, the angles between lines will change. A straight line may become a curved line. But let's say you have a loop that you've drawn. Once you stretch your rubber sheet or you blow up your balloon, it will remain a loop. So that latter kind of property is a topological property. But kind of the notion is that we want to interject prior information, but we don't want to interject it in a very rigid way, because one of the problems with the older, good old-fashioned AI, so to speak, was that the rules that were hand-coded were too rigid. And so this is more an attempt to build in somewhat more flexible rules. So give the computer some guidance, but not very rigidly. So there you see on the left a neuron in the gray square. And if you look at the neuron, you look at the bright um, image tracing of the axons. There is one part where it fades a little bit. Maybe you can put your cursor on the part where it fades a little bit. That's sort of a topographical map of that, interpreting the brightness as a height. You can think of it as a mountain topography. And the neuron then forms a set of mountain ridges. And and you can see that the mountain ridge goes from mountain top down, to a low point on the ridge and climbs back to the mountain top. When it goes down to a low point in the ridge, you might lose signal into some kind of noise background. And the topological technique in this case, something called Morse theory, allows us to construct the mountain ridges, a so-called Morse complex, extracts it, and those mountain ridges then can follow how the neurons are going. So if you go on the bottom panel to the very left, that's the original image, and you can see there's this kind of somewhat faint region, and in the middle, in fact, the human annotators missed that particular connection, and the when we trained a deep net, it also showed similar misses. And when we applied the topological kind of add-on to the deep nets, we were able to capture some of those mountain ridges that were disappearing into the noise. So at some level, this is a small technical detail, but it illustrates how a topological idea could be incorporated into the analysis.
Cool. It's really kind of pulling together math and biology into one very interdisciplinary mix. And you once told me that we were talking about what's it like to do multidisciplinary work, interdisciplinary work. Can you just sort of describe that for other people? Like it's, this may be simple, that may be simple, put it together, it's not so obvious.
Yeah, I think that, of course, this is not just my work. This is work for a whole team. It couldn't be done without that team, and each person has their own expertise. I think what I think of is when people think of multidisciplinary, interdisciplinary work, is that you have a team of experts and they work together. One thing I feel is very crucial is that multiple disciplines need to live inside one person's head. And the reason is that Without that, it's sometimes difficult to quickly make the connections between the different disciplines. I think one person can think faster than a committee can sometimes. There's the wisdom of the crowds, but there is something to be said about interdisciplinarity within your own head. So certainly, I encourage my team to pick up multiple disciplines of people I collaborate with, certainly bridge disciplines that way, and I myself try. And here the application domain is biology. We're doing biological experiments. My colleague Yusu Wang, who is a computational topologist, um, I had a conversation with her at a machine learning meeting several years ago where she was showing me what she'd been doing, and I looked at that and I said, "Oh, that could be applied to our neuroanatomical data analysis." And at that time, we weren't really thinking about deep nets. So Shomik Banerjee, who's the lead author on this paper, has a PhD in machine vision, and had picked up kind of expertise in deep net. So just going across the table, we were able to put together these different pieces.
That's amazing. So one of the things that we were looking at those images before we actually started having the formal conversation. There were a bunch of beautiful images of brains, and each one of them looked kind of different. One of them was the octopus brain with huge optic lobes, and the central brain was clearly not organized the way ours is.
Yeah.
So what's the challenge? And I mean, what would we learn by comparing the anatomy of different kinds of brains? Like, that it seems like there's something very fundamental about our appreciation of those brains, but we don't understand our own. We wouldn't necessarily understand theirs. But what do you think when you look at these different kinds of brains? I mean, what do you think about?
Well, the hope is that we can discover the logic by looking at the wiring diagrams. And there's good evidence that we can do that. If you look at the visual system, for example, I think old example is, in studies of the horseshoe crab, where lateral inhibition in the visual system was perhaps first discovered. You can almost see the algorithm in the circuit. You see how the neurons are connected. And if you understand it well enough, you could think about it almost as a little circuit, a Boolean circuit that implements a particular algorithm. Now, that kind of trick may work better in the sensory periphery. I don't think we quite understand how to do that with more high-level cognitive processing. So I think to make progress, one also needs theoretical constructs. So the theoretical constructs that certainly I try to bring in come from engineering. And so we look at distributed circuits, which implement algorithms, and ask, is there a similarity between that and what we see in biology? There is, that's very interesting, and there's a two-way street. There might not be, and then maybe we can discover new patterns. So that's at least one way of approaching things. If you look at different brains, one can compare. There's the so-called Krog's principle in comparative biology, which says study the specialist. There's an organism that is really specialized at something, maybe it has elaborated out a particular algorithm in its brain, so to speak. And then by studying the specialist, we may see a particular motif that has been emphasized, and then pick up clues. It's detective work. So.
That's interesting. So, I mean, the octopus brain, for example, I mean, that seems to be, because it has so many arms, it has to be specialized in some way to have arms. Is that sort of the kind of, I mean, where do you start to figure out like these rules for how brains must work?
I think, certainly the approach that I find very fruitful is think about building it yourself. So if you were building an octopus, if you're building a person now, we'll get into Mary Shelley territory there. And this is a place where we've been going for a while, right? But that's, I think, very fruitful. So if you start thinking like an engineer about an octopus, well, you certainly know from biology that it's got this very distributed kind of spinal cord-like circuitry in each arm that can give it local control. So trying to mimic the behavior of an octopus arm might help you in an engineering fashion, building it out with the controls might help you understand the logic of how the brain's laid out. That approach has not really been very successful, I would say, for the central brain. So the piece of the brain we were looking at is the central brain. There are more of the mysteries are there. The peripheral nervous system, perhaps we have a little better understanding of, but I take that kind of engineering analogy quite seriously. You can also see how it differs from, let's say, modern machine learning, because if you want the circuit architectures of the deep nets or many other machine learning algorithms, they don't really match, even though they were inspired by uh, animal brains, vertebrate brains, they don't really match when you look in detail.
Hmm. So I mean that's interesting. I mean, on the one hand, we've used the human brain as a pattern to try to build uh, fake brains, computer brains, right? But maybe maybe we need a different animal to analogize. I mean, so we know a lot about the fly brain, for example. I mean, does the fly brain have different rules than the human brain?
So when you say we have used the human brain as a pattern, that is true at the level of kind of cognitive psychology. So, Turing, for example, sat down and tried to imagine how a person being himself solves a math problem. Take a piece of paper, write something down, think about it, write something else down. And he mimicked that process. So all that kind of biomimesis was done at the psychological cognitive level, not at the brain circuit level. At the brain circuit level, you could say that machine vision algorithms have taken, there's been some back and forth there. But we don't even know how the human brain is really connected. So currently we have a big knowledge gap there. I personally think, one really has to study the human brain. Of course, we do study for practical reasons the mouse brain, the fly brain, non-human primates. But I think within our lifetimes, I hope we will have good circuit maps of actual postmortem human brains. That would
Wow,
be exciting.
wow, that would be like
Yeah.
ridiculous. That would be amazing. So if you, I mean, like if you had so we once talked about this. So you're going to be upset with me. So then you can tell them why. We talk about AI as though it's a person. You know, we try to humanify our machines, and you object to that. So why can't we think of our machines as human-like? I mean, when you talk to Siri, you talk to your phone, you talk to, your car. Now you talk to your car. How many more people are now naming their cars because they talk to them? I mean, what's wrong with that?
Well, I mean, of course, everybody is free to uh, offer it as they wish. I mean, I just find it amusing that, and it's a property of humans to attribute, ghosts and spirits and just really personalize everything, ranging from the ancient tree spirits and so on. So we are doing the same thing to the technologies that we are building. Remember the whole Tamagotchi thing? You had this virtual pet.
Yeah, that was
People
weird.
got attached to it. So I don't, per se object to it. Of course, people are free to do what they want. But conceptually, I think uh, it's a bit of a misleading thing to people say an AI, almost as if it were a person. Um, there is no, kind of real similarity between us sentient humans and essentially the computer programs. I remember a famous computer scientist at Bell Labs put it this way. I like it. These are just giant calculators that we are talking about. So every time somebody says an AI, I also want to say byline, giant pocket calculator.
So someone asked a question. This is actually kind of an interesting question. Um, is there research on the difference between, so this person is curious about intelligence
Yeah.
between different species. And they're wondering if at the cellular or molecular level, forget about the connection level, but is it maybe something different in our chemistry and the connections are, I mean, not unimportant, but they're not really where we look for intelligence? Or I'm sure it's
Well, so the question really is that there are other aspects to brain circuitry than the anatomical connectivity. There's the neuromodulatory effects, and certainly when we think of circuit architecture, we include all of that. I would not, although it is convenient to map the anatomical circuitry, we are also then interested in mapping the electrophysiological, the things that govern the electrophysiological properties. But if you were to ask me, and people, also go looking for special cell types, what's the special cell type that humans have that make them special? I think that from the point of view of, and taking a little bit of a leaf from Turing's book, Turing showed this concept of universality, of being able to replicate one Turing machine with another. So if you take that philosophy, it's kind of unlikely that our capabilities reside within individual neurons, the properties of individual neurons. It's really a property of the circuit. So it's a different kind of circuit that helps us learn language easily. That's the one species difference that all our closest cousins, the other 70, 80, 100 hominins are not there anymore except in fossil form. So we can't have any conversations with them. But they might have had, special circuitry that we also have that allows human children to learn this very complex thing called human language quickly. And I don't think it's, really, certainly it's implemented through genomes, but there's going to be a brain circuit that's going to be different. I'm doubtful that it's going to be property of an individual neuron at a biochemical level.
Yeah, so here's another question. Keep them coming, incidentally, people. So if you find the ability to strengthen weak neural connections, like if we know the circuits and we know where there are weaknesses, is that one way to address Alzheimer's or other neurodegenerative diseases? And there's the second part of this question, or is it the pruning? Like if we knew what during development, which cells had to be there and then go away, is that the target that people would, is that one of the reasons why people want to know this?
Oh, certainly. I think that there are medical motivations in understanding the circuitry. Certainly understanding, maladapted or non-neurotypical circuitry. If you don't know what's different, one can't really start addressing it, right? So the first task is to understand what is different between the neurotypical brain and the non-neurotypical brain of different sorts. And you may or may not want to address that difference. Sometimes it might, be leading to things that somebody says, I really want that particular trait. Don't try to take it away from me. But if you do want to address it, and to the extent that it is there, the properties of the circuit, if we know what it is that is causing the difference, then we have an opportunity to either enhance it, or to decrease it. But those are some of, I think, the underlying background motivations. I would say that where we are today, we don't have a good grasp at all of what human brain circuits are like, let alone what goes wrong with them. We are really, knowledge quite primitive. And I'd like to give you an example. So you can, now there are beautiful neuron reconstructions coming out of the mouse brain, single neurons, let's say a motor cortical neuron, something that, your motor cortex is roughly around there. And from the mouse motor cortex, there's a neuron that starts and spreads out branches like a tree, and you can see it, right? Now, nobody has seen a human motor cortical neuron. Nobody, in its entire glory, going down from here, down through your head to your spinal cord. And it's a technological reason why we haven't seen it. But people say, it's like a moonshot going to the moon. I say, well, people have gone to the moon. We haven't yet seen a human motor cortical neuron. So there's a real challenge, right? Somebody wants technological challenge.
That's true. It's
So
true.
we have this big knowledge gap. We don't know how the normal brain is constructed really at this kind of level. We are just, kind of finding out little pieces of information. And then what's happening in the non-neurotypical brain where there's a disorder. If you don't know what the normal is, how do you compare? I'm not, trying to exaggerate. Of course, we know a lot, but at the same time, we know little really.
How would this kind of, it's going to be a very visual sort of machine learning tool that you're building. Um, what other applications does it have besides neuroanatomy?
Well, I mean, the kind of thing we are trying to do is where you have a dense image, an image full of stuff, and where we are trying to label every little bit of the stuff. So that's still a hard problem in machine vision, as we found out. Typically people are saying, well, here's the chair, here's the person, here's the car, here's the tree. But this is more like every leaf of the tree and all the grains of the chair and all the little screws in the chair. So it probably will have other applications in machine vision. One particular one, a germane one, would be in anatomic pathology. So there's been, that's one place where you have physicians looking at glass slides under a microscope and making a diagnosis very quickly. If you watch an expert anatomic pathologist, it's remarkable, and very quickly do the detective work and with their vast store of knowledge tell you what's wrong. And anybody having a biopsy, colonoscopy, a skin biopsy, this is what's happening. And there are a lot of efforts to trying to automate that, but with all those efforts, again, I don't think, and you will hear claims that they are as good as human experts or better than human experts, but no, not really yet.
But there are two of them. There's the melanoma one and the breast cancer one. Those are supposed to be pretty good, aren't they?
Well, again, it's all in the details. So there are particular instances where the FDA has approved, for example, algorithms that will help together with specific stains and marks. But I'm saying in general, there are hundreds of these conditions, and it's a difficult problem. It's not a solved problem.
Yeah, no, I think
I'm very interested in it, but these are kind of currently germane research topics, and it's easy to publish a paper and say, I got this percentage, but are you going to really trust your life to it? Or
that's
so
true.
we haven't quite crossed that bridge yet.
That is true. So someone asked an interesting question of you personally. What is your favorite mathematician and why? Who is your favorite mathematician and why?
My favorite mathematician. Oh, I don't know. I think that we are all fascinated by all these different stories. Uh, I'm reluctant to like name an individual because I would like to say uh, my favorite mathematician could be a child who shows a little spark of mathematical thought that I'm surprised by. Why, so my favorite mathematician is in some sense every kind of creative, imaginative human being. We are all inspired by the famous named mathematicians. As a physicist, Isaac Newton having invented calculus and having had a fight with Leibniz about that, or if you think of you go back and say who is it that really formalized the notion of zero? There's the Indian mathematician Bhaskara. You think of these people, or you think of Pythagoras, or algebra. So you think of the Arabic mathematician who wrote this text. So all these names come to mind, but I really think of the unnamed, the curious child. That would be my favorite mathematician.
That's amazing. That is amazing. So someone asked this, we talked a little bit about it, but I think, it bears repeating. What is the scale of the problem for neuroanatomy? Like you were talking about the number of images in Facebook versus the number of images of brain, but could you just walk people through just very quickly how you get those images? How many slices does it take? And, give people a real sense of the scale.
Yeah, so a photo you take with your camera is a megapixel photo, roughly speaking, maybe 10 megapixels now. A photo we take of the mouse brain or is a gigapixel photo, so about 1,000 times bigger photo. So a little hard to take that photo on your camera. So we've got a pretty expensive camera, costs $250,000 to take that photo. And now you need a even more sophisticated camera to take a picture of, let's say, a whole human brain size. That's a terapixel. Terapixel is like the scale of Google Earth. So you basically have got satellite images, you stick them together, you get terapixel sized images of the Earth. That's basically one human brain slice, one slice. And then depending on how thin, this is at light microscope resolution. And now you take, 10,000 such slices, right? So 10,000 Earths, 10,000 Google Earths, that's like one human brain worth of data. So The scales really are truly enormous. Now, it's not practical. We are academics. We don't have the money that Facebook has or Google has. Sometimes there are collaborations, but so we have to be, careful. We compress data. We have to kind of bring it down to earth, so to speak. But these are large-scale problems that to really understand will take the kind of funding that particle physics has commanded in the past. That's really the scale. These are very large-scale problems. We are scratching the surface.
Well, that's amazing. Somebody else is asking, how many connections? Actually, that's an interesting question too. How many connections are there in a human brain? Because you want to find them all.
Well, that's an interesting question. So first of all, I don't want to put a number on it. People do put numbers on such things. It's astronomical. People will say there's 10 to the 12 neurons. So that's a million neurons, and each neuron maybe talks to thousands of other neurons. So now you can do your calculation. So thousands of trillions of connections, if you want to think of it that way. But I'm not sure I want to think of it just mapping all the connections. People do say that, I'd like to think of it hierarchically. There's organization at the level of, you fall down from a horse like a famous actor and you break an important set of connections between your brain and spinal cord. You don't worry about your million trillion connections. So there's a hierarchy to these connections. Some are, and they're grouped together. So the way we are going about mapping these connections is also kind of hierarchical. So we studied at the so-called mesoscopic scale, where it's kind of regional connectivity. Using electron microscopy, people want to study synaptic connections. And such a hierarchical view is essential. Otherwise, we'd just be staring at our data, it'll become wallpaper.
It's true, that's true. So here's someone else asking about how the circuits change or evolve over time. So those 10,000 slices are a snapshot in time. Pardon?
It's post-mortem, so it's no longer changing anymore. We don't have any method in the human brain, and it's doubtful we will ever have something that will map those circuits in real time at those microscopic scales. How do they change over time? Well, they change very rapidly during development. They also change, during senescence. And in between, there's a region of relative stability. I would say you go to sleep, you wake up, you're still Aline, and you remember. I guess if you're writing one of those books where you're remembering the little cake that your aunt made when you were a child, and so somehow those connections have persevered, or maybe they were maintained. So I think there's, relatively stable patterns, but of course we form memories. So there must be changes going on. I think the thing that really is remarkable, I find, is the fact that different species have different capabilities. We're almost the same. You look at a non-human primate and humans, are almost the same. Yet we have very different capabilities, and those capabilities you can't really teach a non-human primate a human language in the way that you can teach a human child. So there is something, even though we are very different, even though the circuits change, there are some capabilities which are universal or invariant, and not there in the details of the circuitry. They're in some pattern. Those patterns won't change if you're a human, but could be strengthened, could be weakened. So this goes back to the fact that there is a genetic program that's driving all of this and forming us. But within that discipline, of course, there is a lot of plasticity and flexibility. People can teach themselves. I saw amazing, somebody solving a Rubik's Cube with their feet in a matter of seconds. Now, maybe that person's brain is wired in a very, very strange, different way, but I suspect that person has also really, really practiced and practiced and practiced. And that's remarkably changed it too.
That is amazing. So we have a lot of questions coming in. What is the one development in neuroscience that you would love to see in your lifetime?
Well, I think, we really have no understanding of fundamental things. And we think we do, but we don't, is my kind of opinion. Like visual memory, you see somebody, people call it one-shot learning once, and you remember. How does that happen? I think there are these like, in the traditional kind of Skinner Pavlov, you're repeatedly conditioned with some stimulus. In the laboratory, you expose an organism to trials, hundreds of times. But you and I, we see something once, we remember it. I don't think, this kind of mystery is there. So there are a number of such mysteries. I won't lay them all out. But it would be nice to gain insight into one or more of those mysteries. The one that fascinates me most is language. What is special about the human brain circuitry that enables it to learn language?
That would be interesting. So practice. One of the reasons that those pathologists or anatomists are so good is they have the 10,000 hours of practice. So someone is asking about, so you practice one thing and someone else practices another. Do you expect to see a variation in the patterns, even between, like similar enough tasks?
Absolutely.
What's the kind of mapping that you need to do to figure out?
Well, people have been doing that using fMRI. Again, this is the only non-invasive technique that we have. So we can't get down to the very microscopic levels. But there is plenty of evidence of practice-dependent plasticity in the brain. There's a famous, examples of the phantom limbs where Ramachandran essentially caused a virtual surgery by having a person move their phantom limb with a real limb being mirrored, and was able to change the representation in the brain of this phantom limb. I don't know if you know the story, but there's plenty of evidence that that there is plasticity of this sort. I don't think, unfortunately, we will be able to do that at the level of the circuit details that we're talking about, because it is not going to be possible to cut open somebody's brain every time, take out a little sliver, so we're just going to have to speculate about that. It's the same way that we can't rerun evolution, we can't rerun, how gravity pulled together galaxies and stuff like that. So some things will just have to infer.
So people, I think people tend to be fascinated by this. Non-human primates cannot learn language the way we can, but some of them can speak with sign language, and even cats and dogs can use push buttons to communicate. How will any of your work actually help us figure out that sort of fundamental level of communication?
Well, I started, and one of my favorite things is the bird brain, and I studied birdsong for 10 years. And I think we've gained a little better understanding as to how birds mimic, learn, imitate songs, and at the circuit level. So I think yes, and some of those lessons will transfer to humans as well. So, leaving aside language per se, communications between animals is, of course, quite universal. Even plants communicate, of course, at a solar level, not using electrical signals per se. But so yes, I think, and we already have gained, I would say, in model organisms, knowledge about neural circuitry that are involved in communication. So I would not conflate communications with linguistic thought, which is something a little different.
Fair enough. We have time for a couple more questions. So someone asked, what is the ratio of number of neurons between a mouse and a human brain? So what, and actually, I'm curious about
roughly 1,000. I mean, I don't know exactly. Like I said, you can cut out large parts of the brain and nothing happens. So, I mean, nothing happens at the level of, I can still talk to you and you can still do stuff. So, but I would say roughly 1,000.
And what's the simplest circuit that actually, I think what aplesia has a very simple circuit for behavior. I mean, you were talking about engineering a brain. Does that mean that a mouse is 1,000 times dumber, or what does that mean, that ratio?
No, I think that's exactly the point, right? We don't really know how to interpret these numbers. Just because an organism has very few neurons doesn't mean it's compared to, concomitantly less smart because it might be using those neurons in smarter ways because it's forced to. On the other hand, it's very difficult for me to imagine that you don't need the larger brain sizes at some level. So there must be something about the many number of neurons that we have. And one possibility is that it's the same kind of reason, so you can think of robustness. You lose neurons, like cosmic rays or whatever, killing a bunch of neurons each day. You don't want your circuit to be perturbed, so you could get robustness by simply law of averaging, but there may be more sophisticated methods of coding where you gain robustness by having many neurons. There may be a lot of detail. Primates are very visual animals with a lot of brain territory to visual imagery. So to simulate and to remember all those images, you just may need a lot of units.
Yeah, no, interesting. Well, we have to wrap it up here, but I'm going to take the moderator's privilege of asking a question that came up for me. Yeah. And that is when you're talking about these levels of complexity in between different kinds of brains, and like you were pointing out, the octopus brain is has a very complex problem with 8 arms and 8. Would you imagine actually that we could develop better humans if we understood what would make those like, could we in essence have a bionic octopus head? I mean, the interface between us and machines is getting better and better. But if we understood other machines, could we actually add functionality to ourselves just by understanding how those circuits work in other creatures?
Well, people are, kind of trying to build neural prosthetics if you lose functionality. But just taking that as an example, visual cortical implants to give you back a little bit of vision, all you see are, kind of phosphenes, bright glowing dots replacing this really amazing detailed image that we see in front of our eyes. So I don't think in the foreseeable future, there's an amazing, do you know about these twins that are joined at their heads in Canada? They actually have this bridge connecting. So one of the twins can see out of the other twin's eyes. Yeah, and one can move the other one's legs. So they truly are, two brains that are joined at a level where you and I could communicate by language and have connections. But, apart from technology just facilitating that, I don't think we are on the brink of any such enhancement. That's my personal opinion.
Well, Partha, thank you so much for a wonderful and illuminating discussion. I'm going to have that image of the, one twin controlling the other person's hand for a while. You are always such a wealth of big ideas and interesting information. Thank you all on Zoom for your wonderful questions and for your attention and time. If you want more neuroscience and biomathematics research news, check out our website at cshl.edu, sign up for our newsletter, and for now, take care and appreciate your own amazing brain.